thanks to the organizers. Thanks to the organizers for putting together a great event until now. I've really enjoyed it and learned a lot so far. So you've been to, I'm sure, to lots of talks and posters about different aspects of cognition, but as we all know, the motor part of cognition is really the most important part, where we take the thoughts and we express it into our interactions in the real world. And so today we have 5 talks related to the topic of motor control. Throughout the talks, each person will talk for about 15 minutes, and then we'll have a couple of minutes after each talk for questions. If you can save your questions till the end of each talk, and if you can raise your hand, I'll call you. If you don't want to raise your hand, you can also write in the chat, and I'll read out the questions at the end of the session. So we'll start off today with Reut. Thanks.
Everybody sees my screen?
Yes.
Okay. One moment. And everybody sees my cursor? Okay. So first of all, thank you all for coming. And thank you, Jason, for organizing this symposium. I'm Reut. I'm a PhD student, and I'm under the supervision of Dr. Lior Shmuelov from Ben-Gurion University. And I'm also an occupational therapist, and I'm working at the Adi Negev Rehabilitation Center near Ofakim. And today I'm going to present part of my PhD study. So like I told you before, I'm an occupational therapist, and I'm working with neurological patients. And while working with these patients, I got wondering how much the cognitive state influences their motor learning abilities and the motor recovery. And my experience with them led me to feel that the cognitive state is very important for the rehabilitation process in general and motor rehabilitation in particular. And that we therapists are not paying attention to that enough. So I decided that I need to check this out. And in order to do that, I started looking for a task that will test motor learning. And at the same time, I'll be able to also study the underlying learning mechanisms. So a well-known paradigm that fits to what I was looking for is the visual motor adaptation task. Shortly, in this task, the participant is seated at a custom-made table that houses a horizontally mounted LCD screen, which means that the participant doesn't see his hand while performing the task. The participants are instructed to make center-out plantar reaching movements by sliding a pedal on a tablet that's beneath their hand. And during the baseline block, as you can see here, the movement of the participant's hand and the cursor are aligned, synchronized. And during the adaptation block, the direction of the cursor is rotated with respect to the hand, which is called the perturbation. And with time, as you can see here, the participants adapt to the perturbation and succeed in reaching the target. As you can see in this figure I took from Moher and Collins from the study from 2015, this is how adaptation looks like. So we have head angle with respect to trial number. And you can see that when the perturbation is introduced, the participant gradually change their hand direction to adapt the perturbation. And when the perturbation stops, they gradually go back to the baseline mapping. And when the perturbation kicks in again, they adapt again, but as you can see here, much faster than here. So this faster adaptation called the saving phenomena, in which people show faster performance. Performance gain when the same learning task is repeated. So the difference between here and here is the saving. So one interesting phenomena about adaptation that it is driven by both strategic, explicit, and implicit learning mechanisms. We use the same task as Wendell Wurde and JJ from their work in 2019. In this paradigm, you have two kinds of trials. You have cue trials and un-cue trials. The cue trials are trials with perturbation. This is the trials in the learning block and the relearning block. And the un-cue trials are trials without perturbation. And these trials are trials that appear between the cue trials. So these uncued trials appear inside the learning block. And what is the cue? The cue is the cursor. So a change in a cursor color indicates the presence or absence of the perturbation. But what's important to add that the participants doesn't know what happens when the cursors change. They just know that something will happen, they don't know what. So how do we measure explicit and implicit? So explicit adaptation is the difference between activating the strategy and turning it off. So the difference between cue trials and uncued trials. So the participant knows that something now changes and he needs to change his way of doing the task. And the implicit adaptation is the unintentional shift in hand direction in the uncued trials. Like I told you, the trials that appear between the cue trials. So back to our studied population, why they interest me so much. So stroke is the leading cause of complex disability in the world and the second leading cause of death. Almost 17 million people around the world experience their first stroke every year. In Israel, it's approximately 18,000 each year. And stroke causes long-term motor deficits and also cognitive deficits, such as memory, orientation, language, executive function, and more. And motor learning abilities may be impaired following stroke. This topic has a lot of debate in the literature. And our aim was to examine explicit and implicit learning and saving of visual motor perturbation in chronic stroke subjects, age-matched healthy control, and young control group. And why do you want to do that? First of all, we wanted to characterize the effect of cognitive control on motor learning. And second, we wanted to understand the consequences on rehabilitation, which is very important for me as a therapist. So what we had 27 chronic stroke subjects, healthy age-matched control, 31 of them, and 30 young adults. All the participants went through a cognitive assessment. The adult participant, the stroke and the healthy, went through a screening cognitive assessment, the MOCA assessment. And the young participants went through a memory assessment. We took a modified version of the California Verbal Learning Test. In this task, the young participants heard a list of 16 words, then they needed to immediately repeat the words, then they heard the list again, then repeated immediately again. And after doing the motor learning task, they needed to recall the words again without hearing them again. So each participant received 3 scores after each repeat, and the score was the amount of words they remembered correctly. And all the participants went through a visual motor adaptation task. This was our settings. It's similar to JJ, what I showed you before, but a small change in the number of trials. This was our setting in the lab. Thank you for Jason and IKEA for this elegant design. It was their Jason's idea, and thank you. And now let's talk about the results. So our first hypothesis was that stroke subjects will have, first of all, cognitive impairment compared to their control group. And because of that impairment, they'll have impairment in adaptation and saving. So we first found that stroke subjects really had reduced ability, cognitive ability compared to their age-matched healthy control. You can see here the total MOCA score out of 30. 30 is the max score you can get on MOCA. And the groups, you see the difference between the lines. So we did have a difference in the cognitive abilities. But when we looked on adaptation, we saw a different picture. I'll just explain first the graph. Here you see the mean hand angle. This is the learning block and the relearning block. The solid line is the mean of the hand angle in the cue trials, and the asterisks are the performance in the uncued implicit trials. So you can see here that both groups adapted to the perturbation similarly, no difference between the groups. And another nice thing, we can see that we have saving effect. Here, you see the difference between the beginning of the learning block and the beginning of the relearning block. But when we looked on age, we saw a different picture. So we found significant difference of time, group, and interaction in both learning blocks. In the first learning block, the interaction was driven by the fact that the change over time was significantly bigger in the young group in comparison to the other groups. And in the second learning block, the interaction is driven by the fact that the difference through time was significant only for the stroke and healthy, but not for the young group. So we can understand from that maybe young participants managed to maintain the level of adaptation between blocks or adapted quickly in the readaptation blocks. We're looking on saving. This is our saving measure. Here is the mean hand angle in the first trials of the adaptation block and the first trials in the readaptation block for all groups. We can see, first of all, that all groups had saving effect. We can also see that there wasn't a difference in saving effect between stroke and healthy. So stroke did not affect saving effect, but we did receive interaction between group and saving. The difference between the first trials in the adaptation compared to the readaptation block was bigger for the young group compared to the other groups, which should suggest that the saving abilities decreased with age. We're looking on explicit adaptation. So we calculated the explicit component by subtracting head angle direction in the uncued trials from the cued trials that appeared before them. We had eight of those per block. We can see here that the young participant had overall bigger explicit component, and the explicit component increases with time in the first learning block, but not in the second learning block. So this suggests that the explicit learning improves over time, but only in the first block, and then reaches some kind of plateau. And we found no interaction here. When looking on implicit adaptation, we analyzed the data from the uncued trials, and again we had 8 of those per block. When looking here, we saw that you can see that implicit learning did not differ between the groups in both blocks, and furthermore, we found no significant difference between the block and the implicit parameters. So we didn't see an improvement. So the result may suggest that the explicit parameter is more influential by age and has greater contribution to saving, while implicit learning contributes to the adaptation process, but is less influential by age and stroke. So our last hypothesis was that cognitive capacities in stroke in all adults will be correlated with explicit learning in adaptation. So as you can see here, this is the change in explicit adaptation, and the x-axis is the total, the cognitive scores the participants got. This is the MOCA score, and this is the memory test scores. So we found positive correlation between these parameters for the stroke group, and also significant correlation between the scores in the memory test and the change in explicit adaptation. This was interesting for us to find it also in young participants. And to conclude, adaptation is not impaired in stroke compared to their control group, but is impaired compared to the young participants. Impairment is associated with reduced explicit adaptation. Explicit adaptation is correlated with memory performance in all groups, and I think the big conclusion is that cognitive control is important for motor adaptation. And the underline of all of this is that cognitive state of stroke patients should be taken into account in motor rehabilitation process. And I think this is my next step in my PhD study, how to implement this in rehabilitation. And that's it. Thank you for listening. Thank you for my beautiful lab. Thank you for Rotem for helping with the data collection. And just say that this is now a great time to come to the Negev. We have a lot of Kalaniot. You're all welcome to come, and thank you very much for listening. And I think I have time for questions, right, Jason?
Right, yeah, if anyone has questions, please raise your hand. In the meantime, I want to ask you a question. So it looked like the younger participants reached a plateau relatively early and the older ones kept on learning. Do you think if you had given them more trials, they would have got to a similar level at the end?
No, no, I don't think so. The one who didn't, the adults who didn't get it, and I don't know if it will happen. I think it just will cause them to more irritation that they're not succeeding and reach the goal.
Okay, and did you, in regards to the explicit part, did you ask them if they understood what was going on, to see if they could, as well as your explicit measures, if they could verbalize what you saw?
So I didn't ask them explicitly, but I was with them all the time. So some of them did explicitly understand, and some of them did not. But you did see adaptation through time, but it did not understand what's happening explicitly. They did understand that something is happening, but they told me I'm succeeded in the circle, but I'm not succeeding when it's triangle. But they didn't know what is happening. But some of them did. Some of them understood and explicitly knew what happened.
Mm-hmm.
Let me just add that we also tried the aiming task, and it was impossible to implement in stroke and in elderly, because it's raising the attention or the consciousness of subject, of older subject in this task is really hard.
Mm-hmm. This is where you ask them on the clock face where they'll plant, where they.
Yeah, we tried that in the beginning. We had a pilot, and they got really irritated by that. They didn't want to finish the task. They told me, I don't understand what you want from me, and we understand that we needed to change the paradigm.
Mm-hmm. Okay, great. Thanks, Ruth. If you don't have any more questions, we'll move on to the next speaker, Batel.
Yeah,
just close the screen, Ruth.
Yes, just
Thanks.
I'm trying. Okay, I did.
Can you see my screen? Yes,
yes,
great.
Yes.
Just let me see that it works. Yes, great. So hello, my name is Batel. I'm a PhD student at Roy Mukammel's lab. And the project that I'm going to present to you today is examining the interplay between motor and sensory signals and how this interplay affects learning of a sensory motor skill in healthy adults. So in order to perform a successful skill, we need to be able to fully predict and fully control the interplay between our actions and the sensory consequence we want to achieve. And we know from previous studies that voluntary actions modulates our perception. So if I do something actively in order to trigger a sensory stimulus, I perceive this stimulus in a different manner than when I simply perceive it without having any motor engagement. And maybe the most intuitive example for this phenomenon is the fact that we can tickle ourselves. If we try to tickle ourselves, it feels less intense than when someone else is trying to do it. And this effect is not only specific to the tactile modality, but it was found in other modalities as well. So for example, in the auditory modality, it was found that participants' hearing thresholds were lower, so they could detect lower intensity sounds when they were actively triggering the stimuli than relative to when they were passively perceiving the same stimuli. And the theoretical model that's trying to account for this phenomena is called the forward model. What this model is saying is that when we perform an action, we don't only send a signal to our muscles to make them move, but we also send an efference copy or a prediction copy of what we expect is going to happen to relevant sensory regions. Now, this model is a theoretical model. It doesn't say anything about the specific anatomy of the motor system. But given that we know that a motor system is highly lateralized, so the right part of my brain is controlling the left side of my body, it is possible that this kind of signals are also experienced some dependent on the laterality of the action. So we previously found evidence for this phenomenon. So for example, in the visual modality, when we ask participants to compare two visual stimuli, either experienced passively or triggered actively by a button press, and we ask them to report which stimulus is brighter. If we want to quantify the modulation, the perceptual modulation of this stimulus, we want to examine the tendency of them to choose one stimulus over the other, either the active as brighter or the passive as brighter. And in this particular experiment, we manipulated the stimulated visual field and the triggering hand of the stimulus. So it was all combinations of visual field and hand. So right hand, right visual field, right hand, left visual field, and again for the left hand. And we measured their tendency to report one stimulus as brighter over the other. And what we saw is that in the ipsilateral condition, when stimuli were presented ipsilateral to the triggering hand, participants reported more stronger perceptual modulations relative to when the identical stimulus was presented using the contralateral hand. And we find another evidence for this effect looking at brain data. So when participants used either the right hand or the left hand to trigger the same visual stimulus, so they observed the same stimulus, we see a differential activity in visual cortex, depending on if the stimulus was triggered with the right hand or it was triggered with the left hand. So visual cortex cares how the stimulus was triggered. And similar results were also found in the auditory modality. So as I showed you before, if you check the hearing thresholds of participants, hearing thresholds were lower for stimuli that were presented for the ear ipsilateral to the triggering, the stimulus triggering hand, rather than in the ear that is contralateral to the stimulus triggering hand. And also, if you look at neural activity at auditory cortex, for stimuli that are presented binaurally, so in both ears, auditory cortex responds more strongly when it is ipsilateral to the motor cortex that is activating the stimulus. So again, we see a similar effect. So if we try to conceptualize this effect, so if I want to play the piano using my left hand, my motor cortex will have to send a motor signal to the left hand so it will move. And if I expect a binaural feedback, my motor cortex will also send different signals to auditory cortex right and left. And this signal will be stronger within hemisphere relative to signals between hemispheres. And now that we know this relationship, we can try to probe how will it affect motor learning. So if we want to try and learn a sensory motor skill, such as playing the piano, if I want to train my left hand, will it be better if I train with a signal with feedback to my ipsilateral ear, or will it be better if I train with feedback to my contralateral ear? So to answer such questions, we designed the following experiment. So we had two main research questions. The first research question is how the configural relationships between hand and stimulated sensory cortex affects our learning. And given that we will find such learning, we want to know how whether such effects will be generalized beyond the specific scenario of playing with ipsilateral or contralateral feedback. So if I play with my trained hand or my trained hand and receive binaural feedback. So in order to do this, we brought participants to the lab, and we taught them to play a short sequence on a digital piano. So this was the sequence. And we asked them to play the sequence as accurately as they could both by the order of the note, so they had to do the exact order, but also they had to keep a constant interval between each note. We call it an IPI, or interpressed interval, of 300 milliseconds. We had in this experiment 4 experimental groups. One group that trained with the right hand and had feedback to the right ear, and one that trained with right hand and had feedback to the left ear, and vice versa for the left hand. So all possible laterality conditions. Overall, we had 117 participants in this experiment. They were all right-handed participants, and they didn't have any prior piano experience. And we brought the participants for 2 consecutive training days. So within each training day, participants had to repeat the sequence as accurately as they could with one specific hand in each group, depending on the group they were in. So in this example, right hand. And they received auditory feedback only to one ear, in this example, the left ear. And they had to try and improve as much as they could. During the training, they had monitoring of their tempo. So they heard the metronome, and their goal was to keep the sequence within the metronome time. So the entire sequence had to occur between 1 and the second tick of the metronome. So they could receive feedback on how they were performing. Before and after each training, we gave participants an evaluation phase. In this evaluation phase, we asked them to do a similar task. So they had to play the sequence as accurately as they could, but they received feedback for both ears, and they didn't have the metronome. So they didn't have any external monitoring about their time. And we tested this in the trained hand, so for the right hand condition in the right hand, and also in the untrained hand. And we tested this evaluation before training to assess their performance prior to any manipulation. And we also assessed this after training to see whether any training effect will be kept constant when we remove the monaural feeds. So our dependent measures, we call it delta IPI, was the actual difference between each note of participants, so between the first and the second note, relative to 300 milliseconds, which is the goal difference. And we took this in absolute value, so we want to look at the magnitude of the difference, but we don't care if they are too fast or too slow. So for example, if they had to play this sequence, perfect performance would look like this with a constant 300 millisecond difference, but participants are not perfect. And it will a participant's performance would look something like this, for example. So maybe the difference between the first and second note is 350 milliseconds, so delta IPI of 50, and the second and third note is 290, so the difference would be 10 because it's absolute. And we took the absolute value because we wanted to quantify the magnitude of the difference between the perfect performance. So if we didn't look at absolute values, this difference and this difference would be average to perfect performance, would be average to 300 milliseconds. But we can see that here and here, the participant was equally apart in performance. We also removed from analysis any errors that participants did. So for example, if I aimed here to a note that is 4 and I accidentally pressed the note 3, this delta IPI was removed. And we did this in order to avoid any post-error slowing effects or something that might compromise the results. So when we look at the results, so in the x-axis, you can see the training blocks in the 2 days, days 1 and day 2. And in the y-axis, you can see the delta IPI, the difference from perfect performance. So for the group that trained with left hand and had ipsilateral feedback, we can see, first of all, that participants improved over time. So in here, lower is better. Lower delta IPI means lower difference from perfect performance. So first of all, our task is manageable. Participants can learn the task and they improve as the experiment continues. And when we look at the left-hand contralateral condition, we see, first of all, that they begin around the same point. So we don't have any inherent difference between the groups. And they also improve, but they improve more than the group that trained with ipsilateral feedback. So contralateral feedback was better in this condition. When we look at the right-hand group, we see similar pattern of result. So again, we see participants improve over time. So this is the ipsilateral condition, and this is the contralateral condition. So again, you can see that they start around the same point. And over the second day, there is a benefit of contralateral feedback over the ipsilateral feedback. It's a bit lower than in the left hand, but it's still consistent in all time points of the second day. So if we average the results, we look at delta IPI across the first day. We don't see any significant difference between any of our conditions. So in Y is the delta IPI, and on the X is the laterality of the feedback. But on the second day, we see a significant main effect for ipsilateral feedback relative to contralateral feedback. So our first research question was answered. It seems that contralateral feedback during training improves performance better than ipsilateral feedback. And now we want to go to our second question. So will our learning effect will be generalized beyond perceiving the lateralized feedback? So if I want to train to a recital and I want to improve my performance, I will train with contralateral feedback, but will it persist if I go to the recital hall and I want to play the piano and now I will get binaural feedback? So to do this, we had the evaluation phase, and I remind you that participants had a similar task in the evaluation, but they used only one hand and they used one hand and received feedback to both ears, and they didn't have any metronome feedback. So in this evaluation, first we want to examine in the first day. So the first day we didn't see any significant difference between the conditions. So the x-axis here is the hands, and the y is still the data IPI. We see slight advantage for the right hand, but it's not surprising because all our participants were right-handed, but we didn't see any laterality effect in here. On the second day, first of all, note the difference in the scale. So first participants improved from the first and second day. So they were better, but the effect was of benefit of contralateral feedback was consistent in the left hand, even in the evaluation and not only in the training, but we didn't see any significant difference in the right hand in the evaluation. Next, we wanted to examine the similar effect in the untrained hand. So will I have any beneficial value of training with IPSI or contralateral feedback if I want to improve my untrained hand? So in the first day, we can see that there is no significant difference between the groups. So I know it's a bit confusing. So now you can see in blue the left hand condition. So the group that trained with their left hand, and we tested their right hand. And this is the group that trained with their right hand and we tested their left hand. And we see no differences in the first day. In the second day, again, we see improvement. So you can see the reduction in the y-axis. And we can see a similar patterns of results. So in the left hand training groups, so the right hand was better if they trained with contralateral feedback relative to ipsilateral feedback. So to sum, we see that we have better audiomotor learning when participants train with contralateral feedback to the hand they're trying to train. And we see a generalization effect of this. So even if I remove the lateralized feedback, I still see a similar effect on the results, but only when left hand was trained, and not when right hand was trained. So this shows that there is a lateralized relationship between sensory and motor cortices in learning a sensory motor skill. Regarding possible mechanisms, one thing that may account for these results is the fact that different sensory modulations and different predictions may manipulate the sensory motor learning, have interference or enhancement that is different, and the relationship between sensory modulations and learning is still not very established in the literature, and it's something that we are working on in the lab right now. Another possible mechanism that may be consistent with the first is the fact that when we have activation of two hemispheres, we have a more prominent neural activation, or more robust neural activation. And we know from theories of consciousness, for example, that this is essential for better consciousness and better perception, and this might also be beneficial for the learning process. That's it. I want to thank my lab members, especially my advisor, Professor Roy Mukamel, and Adar Deri, that is my equal first author on this paper. And thank you for listening.
Okay, thanks, Batel. I think we have time for a quick question if someone wants to raise their hand. In the meantime, I just want to ask you, when you measured the time between the movements, so did you measure from the start of the movement to the start of the next movement?
Yeah, so it was a digital piano, so it was 0 or 1 response. So when we check when we got the first key press to the beginning of the next key press.
Yeah. So the question I want to ask is about did they play with the gap between the movements? Like, would they play like staccato, like faster movements, or would they play like slow movements with small gaps in between? Was this somehow part of their strategy for getting better at doing the task?
Yes, so it's a good question. We at earlier versions of this experiment, we tried to look at this, and we tried to train them to also keep a constant press time, so the time they keep the press down, and it was very complicated for them. So we avoid doing it in the real experiment because it was too demanding and they didn't manage to learn. But we didn't look at this at the analysis. So we only looked at the difference between the beginning of the note to the beginning of the next note.
Okay. Yeah, because I could imagine that they could learn in one, like one of the things could be improved and not the other one. So maybe they're just improving.
Yeah, that's why the first task was difficult, because we tried to teach them both at once.
I have a quick question. It's not quick, but yes. I can ask this time. So I'm just wondering whether there could be an alternative explanation for the framing of do you really need the feedback framework in order to explain it, or what if can you explain your results using something like the Simon's effect, like the congruency between the size and the special attention effect?
Yeah, so that's a good question, and we also asked this ourselves. So the Simon effect is somehow different because it's a response. So you are slower in responding to stimuli that are contralateral versus ipsilateral to the hand, right?
Or you are faster when there is a congruency between the position.
Yeah. So it's a response effect, and in here we are having the other way around. So they had to trigger the stimuli. So it's not exactly the same effect.
Okay. Thanks, Patel. We'll move on to the next speaker, Inbal. Yeah, we can see it. It's a bit hard to hear you, but we can see the screen.
Oh, really?
Okay, that's better now. That's better now. Yeah.
Okay. So yeah, hi, I'm Inbal. I'm very happy to tell you about our research on motor impairment in stroke patients. So this is a study that's done in collaboration with Dr. Ahmed Arak from UCLA and Professor John Krakauer from Johns Hopkins University. It's done as part of my PhD thesis in Dr. Lior Shmuelov's lab in Ben Gurion University, and it takes place in Adi Negev Rehabilitation Village. And the aim of this study was to explore the contribution of interjoint coordination to motor impairments in the early stages of stroke recovery. And as Raut said, stroke is the leading cause of long-term disability in the Western world and the second cause of death.
It's a disruption. It's a condition caused by a disruption in blood supply to the brain, can be caused by either hemorrhage or blockage. Both of the cases can lead to neuronal cell death, which is what you can see here in this brain scan. About 80% of patients will experience some form of motor impairment, usually in the form of weakness and or loss of voluntary movement in one side of the body. And about between 50 to 60% will still have that motor impairment in the upper limb, even 6 months after the stroke. And when we think about this motor impairment in the upper limb that we see after stroke, we can actually attribute it to a number of factors. For example, many patients who had a stroke will report they experience weakness or difficulty using strength while trying to execute voluntary movements. They will also often display lower levels of accuracy and lower or reduced movement smoothness, which is a measure we have for the quality of movement or motor control. These are things we refer to as under the umbrella of dexterity. Patients will also experience impaired sensation or a phenomena called spasticity, which is an increase in muscle stiffness that increases with the velocity of passive stretch. And lastly, they would also often display abnormal coordination patterns. And these abnormal coordination patterns we often refer to as flexor or extensor synergies. And these are actually coupled multi-joint movements of shoulder flexion, shoulder abduction, or elbow flexion. This is in the case of flexor synergy, or shoulder extension, elbow extension, and shoulder adduction. And this is in the case of extensor synergies. And due to these synergies, patients can execute tasks with coupled joint movements that are perhaps inappropriate for the completion of the task goal. Let's think about a patient who is trying to reach an item on a shelf. So that would involve the lifting of the shoulder, but it could also involve the involuntary movement of the elbow bending also. So these are phenomena caused by these sort of flexor and extensor synergies. And this is what has led us to our research question, which is how are these interjoint coordination patterns contributing to motor impairment after stroke, and are they caused by the intrusion of pathological synergies, like flexor synergies that I've mentioned. So we chose to answer that question by utilizing a novel approach, which allowed us to extract 3D model of the human body. This is what you can see here, hopefully. And this is actually based on an algorithm called deep behavior, which Dr. Rak helped develop. And it's based on an algorithm called OpenPose, to whoever of you that knows it. And we extract it actually based on a system, two-camera system. This is what you can see here. And these are high-resolution cameras that we're using to record our patients. Each camera has a different angle. And that's basically what is allowing us to extract this 3D model. And we use the system and to record different motor tasks. These are the tasks I'll discuss today. In the top row, you can actually see the task cup to mouth task. You can see here, this is Reut, if you recognize. She's recorded from two angles. This is the camera input. And this is the 3D position we get from extracting this 3D model. And these are the angles. And if you can see here, this task actually requires the subject to flex the shoulder and flex the elbow in order to lift the cup towards the mouth. This is what Ruth is doing here. And this is one of the tasks. And the other one is a reaching task. The subject was required to reach his hand towards a suspended target. And this required him to perform shoulder flexion and elbow extension. And I hope you maybe recognize that one of these tasks required flexion and deflection, which is within the flexor synergy I've discussed, while the other task, the reaching task, required movement outside of it. So we recorded these tasks on 28 stroke patients in the subacute stage. This is roughly 2 to 12 weeks after the stroke. And you can see here some of these parameters. We also collected an array of clinical measures. We've collected a measure for spasticity used in the clinic. This is the modified Ashworth scale you can see here. We've also recorded a measure of strength using a grip dynamometer. Also, we've tested them on a Fugger-Meyer test. And the Fugger-Meyer's clinical test meant to assess motor impairment in stroke patients was actually developed in order to assess the control a patient has on individual joints outside of these synergies I've told you about. And we also have a measure for sensation, which is a subscale of the same Fugger-Meyer. Also, what you'll see next is we also recorded 16 age-matched controls, healthy controls. And the first thing we did was take this 3D model and calculate different measures of movements, performance on these patients and controls. And this is what you can see here. We've started with basic measures like movement duration and peak velocity. Here you can see these measures for the control groups. Each triangle is representative of a single subject. Compared to the non-paretic arm of the stroke patients, the non-paretic arm is the less affected side. And this is compared to the paretic side, which is the impaired side. And you can see that these values for both tasks. This is a cup to mouth, this is a reaching. And in addition to movement duration, we've calculated peak velocity. And another performance measure, which is extent. We've calculated this by the 3D position of the wrist at the end of each movement compared to the starting position. This is we consider a performance measure for the task. And another performance measure is smoothness, which I've mentioned is a known measure in kinematics. And as you can see here, for actually all of these measures, there was very significant differences between groups. All of the ANOVAs here are significant. Also, when you directly compare the controls to the paretic side of the stroke subjects, this is significant. And also what you can see here is that there's a difference between the tasks, and mostly for most of these measures, the between group differences were larger for the reaching task. That in the reaching test, just to remind you, they had to perform movement outside of their flexor synergy that might be affecting them. So this was one stage of the analysis. We see we have a good performance measure for these tasks. And in the next stage, we took this extent and smoothness and compared them to different types of impairments. So this is what you have in the y-axis, the extent in the reaching task, in the cup to mouth, again, the smoothness in both tasks. And here we display the correlations of these measures to sensation measure, the spasticity, strength, and also a measure for interjoint coordination. And the interjoint coordination is a measure we've calculated on the kinematic 3D data as the Pearson correlation between the shoulder flexion angle and the elbow extension angle. So we have that Pearson correlation measure per participant. We've implemented a Fisher transformation here, so the distribution was a little less uniform. And what you can see here is that for the extent, the significant correlations were with the strength measure and also with the interjoint coordination. This was also replicated in a regression analysis that showed that the most significant contributor as a dependent variable is the coordination measures. And for smoothness, however, you can see actually that only for the reaching task, the strength was significantly correlated. So we take this interjoint coordination measure, and we wanted to explore the underlying sources of what is actually this variance is based on. And the first thing we did was compare it to the same Fugl-Meyer score I've told you about. And so this is the interjoint coordination in the y-axis, and the x is the Fugl-Meyer. And you can see, interestingly, that only for the task that required movement outside the flexor synergy, this was significantly correlated. And the next stage, we wanted to see if this maybe is due to the intrusion of pathological synergies. And we did that using two types of analysis. First, we came up with a measure for the flexor synergies. So we looked at the percentage out of the movement duration in which the subject performed both shoulder flexion and elbow flexion. And this is what you can see here on the left. And quite nicely, we saw there was a difference between the tasks. The task in which they performed flexor synergy, these were relatively high values, and the task requiring movement outside that synergy, the percentages were lower as expected, but there maybe you can also see this, there's very little to almost no differences between groups. Here we also ran ANOVAs, which were non-significant. And another complementary analysis was we calculated the same interjoint coordination, but specifically for the segments in which the shoulder was flexed. Again, here we saw a nice separation between tasks, but no group differences. So hopefully I've convinced you that interjoint coordination does affect performance measures very significantly. However, when we look at abnormal synergies, they have little to no effect on these interjoint coordination deficits. And this perhaps may be indicating that the Fugl-Meyer is not driven by the intrusion of pathological synergies and may instead reflect manifestation of negative signs like weakness or the loss of movement control. And this is me. I'd like to thank everyone who contributed to the study, and Professor Dr. Arak and Lior and everyone in my lab. And thank you for listening. If you have questions.
Okay, great. Thanks, Imbar. If someone has a question, then you can feel free to raise your hand or just ask. I think I might have missed what the definition of the interjoint coordination measure was. If you could just go over it one more time.
How we calculated it?
Yeah.
Just using a Pearson correlation, we took actually maybe we took this angles of the shoulder flexion and elbow extension, and we ran just a Pearson correlation on these two signals, one of the two angles.
Mm-hmm, mm-hmm. Okay.
And so this is like a basic measure of coordination we chose.
Mm-hmm. And have you tried doing trying to look for synergies in the like the usual definition of like using a non-negative matrix factorization or some other technique, principal component analysis, to find synergies in the data to see if that's one of the reasons why you see the differences between different groups.
Are you talking about more complex analysis models? I think
just multi-joint synergies, like time varying multi-joint synergies, if that's part of the reason.
We started doing things like that, but we need to further explore that to know if there is anything interesting. Then I'm sure there is, but we actually, the simplistic approach is also beneficial to us now. But yeah, I think we'll get there soon.
So I think if someone wants to ask a general question, you can, have some kind of discussion if you want. You still have time.
I'll keep asking questions if no one else. Just what would you say, like, in terms of rehabilitation recommendations that come from what you found so far?
Yeah, so we're actually doing things that are related to that now.
Okay, looks like the answer was too good to share with all of us. So
Yes, Leo, maybe you want to add something, or?
Leo, do you want to add something to that?
I think, what we found surprising, but I get it's more relevant maybe to the clinical world, is the fact that, like, Fugelmeyer in Bar is back. Like, Fugelmeyer was considered to be a manifestation of pathological synergies, the flexor and extensor synergies, and that was the main source of impairment. And what Inbar finds is that Fugelmeyer is indeed impaired, but we see impairment in Fugelmeyer, but without indication of pathological synergies, meaning that maybe control problems are still much more are contributing to the motor impairment, and not necessarily intrusion of pathological synergies, and that would affect the way that clinician may rehabilitate the patients. That's an interesting data-driven observation, I think.
Definitely. Okay. Yeah. Thanks, Hilda. And thanks again, Inbar. Glad you're back. Okay. So I'm going to present the next talk. Let me just share my screen. Okay. So today I'm going to be talking to you. About the online and offline effects of changing movement, timing, variability during training on a finger or position task. So I'll talk give a little bit of background about modal learning, just to add on what we've heard so far, and then I'll talk about the particular experiment we did looking at trying to change the variance in a modal learning task and to see what its effects are. So modal learning is a process of learning to perform a motor task in a qualitatively better way. And in general, I think we can probably claim that motor learning requires learning new strategies performing a task. So it's not just, there's always some argument about whether adaptation, is that considered motor learning, or is that something different? But in general, I think if something is a real motor learning, then we have to learn some new sort of strategy for performing a task. And then there are lots of different approaches to this. One approach that we use in a lot of our studies is that we say that when people perform complex movements, these complex movements may be constructed from the combination of a number of motor primitives. I'll give some examples of this shortly. And if this is the case, then motor learning, we can think of motor learning in two ways. One way it can be changes in the way these primitives, these motor programs are combined, or the other way is generation of new new motor primitives or new motor programs. And the difference in these two things is something we probably see in terms of timescale. So it seems like in relatively fast learning in a particular session, it seems like people are only able to do the second option, change how they combine, maybe overlapping motor primitives or the way that they're executed, and generating new motor primitives seems to be something that requires much longer timescales and probably sleep and consolidation along the way. Now, the notion of speeding up motor learning is an attractive notion for many reasons. It can allow us, we never have enough time to do anything these days, so it can allow us to learn or improve our skills faster, or just to learn more in the same amount of time. If we're in like an in-bars talk, if we're in a rehabilitation setting and you have 1 hour with a physical therapist, occupational therapist, so the more you can learn in that hour, of course, the better. And there are a number of ways of speeding up motor learning. Lots of people have looked at this topic in the past. And it's also important to identify the opposite. There are some techniques we know that slow down motor learning, for example, types of interference. So it's also important to understand what techniques don't work or have a negative influence, so we can try and avoid them when we're doing motor learning or rehabilitation. So as I said, there's been many attempts. This is a partial list just to give you an idea of what sort of things people have tried before. There's the big question about how much training is necessary. In the study before, we had looked at how many, whether 100, 200 or 300 trials are necessary. This is always an interesting question. Is it better to mass the training all together in one block or to spread it out over different sessions? If you have more training, does that lead to better retention? This is a study from some of the people at the conference here. Is too much training can sometimes be detrimental as well. And also this is some other researchers from Tel Aviv. We know that memory reactivation, just performing a very small amount of reactivating the memory rather than performing a large amount of training each day, in some cases may be as good or nearly as good as doing additional training. And another method that's being used often is the idea of reward. Is reward a good way to speed up the process of motor learning? And finally, non-invasive brain stimulation, things like transcranial direct current stimulation or TMS. Some of them have shown promise for enhancing motor learning. So I'll just talk to you briefly about the two studies we've done previously in looking at this idea of trying to speed up motor learning. This task is based on the task of Sosnick et al. From in Tamar Flasher's lab. And this task here is to connect the dots in order. What we have to do is join from A to B to C to D and back to A, and do it as fast and accurately as possible. So it's an easy task. We usually do it on a graphics tablet so we can record the kinematics. And in this experiment we had 4 groups. We had 2 groups that observed an expert performing the task. This was someone, she spent 12 days practicing this task, as opposed to just seeing random motion or just having double the amount of training. And then we did the similar protocol to some of the other experiments that have been presented in this session, a pre-test, post-test, and then we looked at consolidation after 24 hours. So in the pre-test, what happens is everyone basically does the same thing. They just join, it's a dot-to-dot task, they just join the 4 dots with the straight lines. And now I'm going to spoil this task for all of you. None of you will be able to be subjects, unfortunately, in my experiments. So the best way of doing this task, instead of drawing 4 straight lines, is to do 2 curved lines, to go from A through B and not stop at B, but make a curved line through B to C, and then a curved line from C back to A through D. And by making curved lines and not stopping, you're actually able to perform the task faster. And so we can see in this task, by this learning by imitation, the 2 groups that saw the observation, the difference here was just that one was slower than the other. See, the groups that observed it were able to perform this much better, and the groups that didn't perform it, that didn't see this, they didn't really improve, or they hardly improved over the rest of the session. And we can see in this case that the difference between the groups, the ones who did observe and the ones who didn't observe, we can see that in the velocity profiles. At the beginning, the velocity in all the groups goes to 0 basically at each dot. So you more or less have to stop when you hit one of the points. And you can see after the training that they no longer stop at these 2 points in the middle. The one difference we can see between the expert and the ones who observed it is that in this case, we can still clearly see in terms of this still looks like 2 movements that are overlapping each other. in both of these cases, whereas in the expert, it seems like she's managed to merge these two movements into one single movement. So we have some ideas that in this case, the expert actually generated a new motor synergy, whereas the people who are learning on a single day aren't able to generate this new motor synergy, and so do it in just overlap the two movements. We had a similar result using the same protocol, but instead of using learning by observation, we did learning using analogies. This was a study of master student, Oriane Zaks. And so in this experiment, rather than showing them what to do, she told them an instruction. She told them, imagine you're painting with a paintbrush and try not to leave paint stains during the drawing. This is what they were instructed to do to draw it. And we had another group, we had a control group with no instructions, and an explicit group where we told them explicitly to make curved lines between the points. And you can see again, as in the control group as before, there's no real change from the pre-test to post-test. And in that analogy group and the explicit group are actually a little bit better than the analogy group, but both of these groups, they're also able to speed up the motor learning process by giving them these hints. So the study I want to talk to you about, this new study I want to talk to you about today is study looking at the role of task-relevant variance in motor learning. I'm surprised that no one actually in this session has presented this exact task yet, but I know Avikani talked about it yesterday in his talk. So any of you who are not familiar, the finger opposition task is where you have to touch the thumb to one of the fingers in a particular sequence. And we use this task because it's been used so many times in the past, and we know that it's easy for people to learn. It's been used to show many interesting findings. For example, the importance of sleeping consolidation, the effective interference from a study we ran a few years ago, that if you learn a different task immediately after learning the first one, it ruins the consolidation of the first task. The effects of time of day of learning in elderly people and ADHD. So if you learn a task in the evening, you'll have better offline learning rather than if you learn it in the morning, probably also due to some sort of interference. Learning after traumatic brain injury, learning by observation without physical training. So it's been used for a variety of tasks, and we often see interesting effects. That's why we chose to use it in this study. And the particular question we were interested in was the effect of variance on motor learning. So this is from a review from a few years ago on motor learning, and talking about the different stages in motor learning. So at the beginning, we have a more exploration stage where we don't know how to do the task in the best way. And so we try lots of different ways of doing the task. So we expect to have, which in terms of variance, we expect to have a lot of variance at the beginning. And then once we get better at the task, we get to the exploitation phase where we know how to do the task. And so we just have much less variance and we focus on doing the task well. We don't have no variance, but less variance at this stage. So there are some previous studies have shown that in healthy subjects that having greater variability at the beginning and the early stages might lead to increased learning. Based on this idea that if you have more variability, you're more exploring the space. And so that because of that, you might be able to learn more. There are some effects that maybe it'll affect retention. There's also been, the evidence is somewhat mixed. Not all the studies that have been done have found a positive effect of variance on motor learning. It's important to note here that I said in healthy subjects, obviously in some motor disorders, for example, if you looked at patients with stroke or people with Parkinson's disease, they'll have a lot of variance due to the disease. So this large amount of variance we know doesn't help them in motor learning. So this is more specifically in healthy subjects, it seems like more variance may help them improve in this task. There is, however, a confound in some of these studies based on what we've seen from our previous works. And the confound might be that if the subjects that have a lot of variability initially may also just be subjects who perform relatively poorly. And so because they're not very good to start with, and because they're not very good, they have a lot of variability. So then they're able to learn more. So we know the subjects that are bad in general, they have more room to improve. Often there's some sort of a ceiling effect in these type of experiments. And this is what we found in the previous study. We compared the variation at the beginning in the pre-test to how much they improved, and we saw some moderate correlation between the two. So the participants who had more variability at the beginning showed more improvement. But the reason for why these same participants that had a lot of variability also weren't very good to start with, which is probably why they were able to improve more as a function of time. So to try and get around this particular confound, instead of looking at the natural variability of participants, we instead manipulated the amount of variance in the within sequence timing. And so the within sequence timing is how long between the touches. And we chose this particular component because we know from previous studies, this is the area that shows the most improvement over learning. And the way we did this is by providing cues for when to touch the fingers. We attached some motion capture sensors to the fingers while they were doing this task. And we had 3 groups. One group was the variable group. Each time they had a series of beeps and they have to copy that rhythm. So here, for example, beep, 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 beep. And then they have to do the same timing with the finger. Each time a different rhythm. The fixed group received the same rhythm each time, the same fixed rhythm. Beep, 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 beep. And they have to do the same movement each time. And the control group would just hear a single beep and then perform the sequence, beep. And so the pre-test and the post-test is the same for all the groups. Just do 4 tests, 30 seconds as fast and accurately as possible. Only during the training is it different, and all the groups have exactly the same number of trials, 10 blocks of the 16 sequences. And we brought them back on the second day as well to check for consolidation. So here's the results. The first thing we saw that we measured, as we used the reaction time, how long after the beep they started moving as a rough measure of cognitive load. And we can see that in the variable one, where it was different every time, we can see that the reaction time is much slower. So it's around 800 milliseconds compared to the fixed, which is about 500 milliseconds, and the control group, which is much faster, about 300 milliseconds. So it does seem that that as well as increasing variability, it does also increase the cognitive load. The other thing we saw is that we saw a main effect of the session as we, in terms of the variability. So we saw that at 24 hours, they're less variable than on the first day, as we expect. And we also saw the interaction of the session in the group, as we'd expect. So only during the training, we saw that our intervention did work. So during the training, they were indeed more variable. The variable group and the fixed group were less variable, and the control group was somewhere in the middle. So now the big question, did it help? Okay, so this is the graph of the improvement. This is the number of sequences they perform. Because there's a lot of variability within each of the groups, we instead look at the improvement of each group. So we saw there's an improvement here. You can see they're greater than 0. So all the groups improved. There's also a consolidation effect. They were better at 24 hours compared to post-test. However, the only main effect we saw in terms of the group was the difference between the fixed and the control group. So the control group actually performed the best, significantly better than the fixed group. The variable group here was somewhere in the middle. They weren't significantly different than the others, but the important thing to take from this is that the variable group, where we increased the variable training, they weren't better than either of the other 2 groups. So how did this happen? They were more variable and they didn't increase. We think that it comes down to where they channeled their variance during this study. So if we look at the component that we wanted there to be variance was the time between when you touch the movements, because this is where we know most of the improvement comes from. And you can see that the only group that actually had more variance in terms of that, or had the most amount of variance, was actually the control group, and not the variable group. So how did it happen they didn't have the variance here? If you look at the other components, we can see they actually had much more variance in terms of the movement time. So the movement time is how long your fingers move for. So the way that they achieved, the way they managed to copy this pattern, beep, 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 instead of by changing when the fingers start moving, the way they changed it was by changing how long the fingers move for. And the unfortunate thing is that when we look at how people improve, the red in here is the movement time. You can see that the movement time from pre-test throughout training and post-test, it almost doesn't change. In fact, sometimes it gets even a little bit higher, even in the groups that do really well. So this component, although they had more variance, this component doesn't actually help us in terms of the learning. You can see that most of the learning actually takes place in all of the groups in terms of this green measure, which is the intermovement time, how long it is between the touches of the fingers. So it appears that the reason why this increase in variability maybe didn't help them is because they channeled this increase of variability into the movement time rather than into the intermovement interval. So in this study, we found an increase in variance in the touch time was insufficient for improving performance, probably because they increased the variance in the wrong part of the movement, or at least where we would have preferred them to increase it in another area. And prescribing a fixed rhythm during the training produced much worse performance, and significantly worse than the control group. And this is similar to what we found in a previous study, looking at self-generated versus cued training. So it seems in these sort of tasks, it's better to let the participants perform at their natural, with their natural timing rather than giving them a specific timing pattern to perform. And it seems to improve their performance. So there are many factors that can affect motor learning in a positive and negative way. Here we showed that the self-paced learning process appears to result in larger gains than using cued learning. And we suggest that these results should be tested in broader populations, not just in young adults, to see how what the effect of this is in other groups. And so, yeah. Finally, just thanks to my collaborator on this and many other studies, Dr. Maria Corman, and to Asaf, who was the student who tirelessly ran the experiments. Thank you. Okay, do that. Okay, if you have questions, feel free to ask.
You have to set the order of those who answer because you handle the discussion.
Ah, okay. Do you want to go first if you're already go?
Yes, thank you. First of all, I did studies which are much related to the studies that you do. We work, I think you saw in the poster on the graphomotor tasks for children. Regarding different conditions and of training and of generalization. Ganamat that you cited earlier in the 2020 study, we just published the paper in JECP. It's also Journal of Experimental Child Psychology. It's also about children, but it's about generalization, different training regimes and generalization. And so I think that the important result is that although one training may be good for the task itself, other types of training are better for generalization. And we show that in the paper and various generalization tasks. And regarding the second task that you did, I think I have a paper from 2008 on the same task, but I think using keyboarding with Avi and Eli Vakil. And we studied, first of all, we studied it individually. So we looked at the individual patterns and they may show, different things. And what was especially important is that when you look at the individual patterns, the learning doesn't look smooth. It looks more in jumps. And the reason I'm saying it is that it has something to do with variability. And also, when subjects perform this task, what they do is that they sort of learn the quantile. I mean, the 5 movements, and the relative gap between those 5 movements is increased, while the gap within the movements decreases. So it's giving them to do it in the same in a constant pace is like telling them, please do not learn the task, because this is what happens when you learn. You amalgamate the unit, and it is different, and you see different units in performance. And this is basically how, at least when you keyboard the task, this is how it's learned. So this is what we showed in the 2008 paper. So just great work. It was interesting to learn more about it. And yes, I think that the different training and consolidation and are still important questions. How do we learn? How can we learn better? And that your studies, they add more to it. And so it's good. Thank you. Thank you for your talk.
Great, thank you.
Okay.
I'm not sure if there was a question there at the end that I need to answer, or just.
I mean, I don't know if you studied also these issues of the difference between what's the best practice for learning, what's the best practice for generalization. In practice, you need to know what is the task that you want to achieve at the end in order to find the best. Okay.
Right. Yeah, we look at transfer as well. I mean, obviously in these tasks, that's probably not such a necessarily a long-term measure of generalization. Okay, I don't want to take over Sigal's time as the last speaker, so I'll pass over to Sigal. I think she had a question as well, but if you want to.
I had a short question regarding. So you talked about timing the period between the finger closure. So what alternative timing mechanism?
Yeah, so the time, because we divide the time into 3 parts: time to move the finger, the time when they touch the 2 fingers, and then the gap between when they start the movements. And it's this gap between when they start the movements, this is where all the learning takes place. So it seemed like we initially thought you should just move your fingers faster and that you'd do better, but that's not how people get better at this task. It's the relative timing is where all the improvement takes place. Mm-hmm. Okay, so pleased to invite the last speaker, Sigal Merman. Thanks.
Thank you, and I'll share my screen. I'm going to share the computer screen because I have some videos I hope to be able to show. So today I'll talk about the individualized joint error treatment for patients with stroke. And we already heard today, we have to do this. Oh, if I do this, I don't see you. Huh? Right? Okay, now I see. So we've heard already 2 great talks today about post-stroke rehabilitation, and that most individuals with stroke have some form of upper limb sensory motor deficit, and stroke is among the leading causes of long-term motor disability. And so treatment today, rehabilitation treatment today is disappointing in that effect. And actually there's even questions regarding is rehabilitation treatment improving more than would have been improved with the spontaneous biological recovery. Now, current rehabilitation practice in general is about high dosage repetitive training. And that means that you have improvement, especially when we talk about the upper limb, we have improvement in practice trajectories and also in practice regions. But with upper limb movement, this is because of the large variability of movement, this is not enough. So the question is, can we do better? Now, a quick jump to reference control theory and to tie in what I'll talk to later in this talk. So we have the muscle stretch reflex, and you see here a cartoon of the muscle stretch reflex arc. And this is basically when the muscle contracts following a stretch that is felt by the muscle spindle. So with the point of the muscle stretch reflex is to keep the muscle at some predetermined length. And reference control theory is about tying in a voluntary motor control with the regulation of muscle stretch reflex. And with the healthy population, we are able to regulate the threshold for the onset of the muscle stretch reflex beyond the biomechanical range, and therefore our active zone, meaning the zone in which we are capable of normal muscle activation, includes the entire biomechanical range. But as we say, however, following stroke, the active control zone is reduced. And this is some images from the work of Malik et al., showing that when elbow, here, this is a passive elbow extension, and we can see that at some point, here, I'll take my annotator spotlight, at some angle, the flexors start activating, and there's an angle for one flexor, and then there's a different angle for the second flexor depicted. The control zone of this patient with stroke is smaller than the biomechanical range. So, okay, so the active zone is reduced. Perhaps we can train to increase the active control zone. And then, of course, assuming that we can, perhaps this would lead to functional improvement. So one of our first attempts in going into this direction was in the enhanced clinical trial that started way back before the Corona days in 2015. And the basic hypothesis was that we would train inside the control zone only while applying transcranial direct current stimulation. And this may lead to natural extension of the active control zone without the patients acquiring abnormal motor compensation strategies. And this training is done in the subacute phase for taking advantage of the plasticity of the brain during this period. So this would be very close to a stroke onset. So this was a tri-national project with India, John Solomon from India, head by Mindy Levine from Canada, and Dari Lieberman from Tel Aviv University, and was done in the Soroka Rehabilitation Center with Sylvie Frankel and Nahum Soroka. And we measured we estimated, so the control zone is the stress reflex threshold is modulated by velocity, and we estimated the zone for each patient using the TSRT measure, the tonic stress reflex threshold, which is estimated based on tonic, meaning the stretch. It's a theoretical threshold at zero velocity, and it's estimated based on measuring muscle activation at different velocities at different at the passive angle, different passive stretch angles. So the patients were their motion was constrained based on the TSRT measurement at each training epoch, and they trained for 2 weeks. They had 5 training meetings, each lasted 1 hour, and during the training, so they received TDCS. That was one group, and then there was a group that trained without TDCS, and then there was a group that trained without this restriction of the elbow joint movement. Now we are currently analyzing the results of this experiment. I will show you, so I will not show you all the results. I will show you results of one measure. The subject, the patients were trained with virtual reality games, and then they were tested with reaching towards 4 target cones. And these are traces of elbow motion during reaching. And what we were trying to do is look at the similarity of motion of patients with stroke to motion of healthy controls. And we use stochastic modeling for that. So we prepare a model, a distribution of the motion of the patient, and we compare this distribution to distribution of the motion of healthy control. And we can measure the distance. And this is an example of Hellinger's distance, which shows the separability of the 2 models. And we have very clear indications that, and for Hellinger's distance, lower is better because you want to be similar to the control. And for elbow extension, shoulder adduction, shoulder flexion, training within the control zone is significantly better, and it's than training outside of the control zone. And you can see the improvement. See, here is the before training, after training, and 1 month after training. And you can see that the patients that trained in the zone continue to improve. TDCS is more fishy. Only we see some effect for on shoulder flexion. So I'm running out of time, but I will say that enhanced analysis does point is in line with the original hypothesis. Second trial, Jason, somebody talking. Thanks. Okay, so second trial, we aimed at taking these thoughts a step further and perhaps training in a way that would lead to dynamic remapping of the muscle level control mechanism. We want to remap so that the active control zone is extended. Just a short, you know what, I'll jump over this slide, but what we're doing, we're adding an error at the joint level, and there's quite a lot of work on adding error at the task level for in training of subjects with stroke. There were promising results with error augmentation for the lower limb, but once again, there is results for the upper limb are disappointing, and we hypothesize that this has to do with the fact that this augmenting of error at the task level mainly leads to more training in task space. So it's like high dosage repetitive training. So I'll try to go through this slide. This is a bit complicated, but this is the main explanation for why we think that adding error at the joint level can assist in extending the control zone of the participant. And we are subtracting 10 degrees. So hypo error. We're subtracting 10 degrees from the angle that of that the subject is from the elbow angle. So here, I'll show it here with the annotator. So if this is the actual arm of the participant, this would be the arm that the participant is presented on the screen. So if this is the original posture in which the participant started, and this is the target that the participant has to reach. So when the participant actually reaches the target, the hand is presented still away from the target, and only after the participant extend the hand further from the target, the participant would see the arm actually reaching the target. The idea is that if the participant indeed is present in this virtual scene, so there will be a remapping of the torque angle characteristic curves on which the muscle is operating, meaning that muscle activation will start in the antagonist muscle only later in the movement. That is, the subject will be able to extend the hand further without interruption of the antagonist muscle. So these are the videos. Here we start out by letting the subject map the region where the subject can reach. This is there's a Kinect camera that's following my arm here, and the arm is situated on a passive manipulator for support against gravity. And then following this mapping, so the zone is suitable for the capabilities of the subject, then the targets appear on the screen, and the participant extends the hand to reach the target. So we did two experiments with this system. One experiment with healthy controls to verify the method, and then we did an experiment with 12 participants in Soroka first ever stroke. They had some tonus in their muscle, so the modified Ashwood scale was more than 0, and 6 participants trained with error and 6 without. With the control subjects in the lab, we saw an effect. We looked at success rate and at duration, time to reach the target, and the training took 30 minutes, and we saw an improvement in the success rate between start of the trial and end of the trial. And we also saw, when we later tested subjects that trained with error, we tested them with a system when there was no error presented, we saw that they took longer to reach the target. We also used the NASA TLX questionnaire to look at mental load and look at discomfort and look and feel effects. It's a questionnaire, it's a 5-point Likert scale questionnaire, and there was no difference between the systems. With the pilot experiment in the Soroka Medical Center, we did not see any difference between the group that trained with the error or without the error, although we saw a clear difference in the Fugl-Meyer measure. Here again, we tested before the experiment and after the experiment, and we saw significant increase in the patient's Fugl-Meyer score. One thing, however, we did not see any differences in when we looked at their training sessions, at their speed and success rate. We saw no difference in between sessions. Here in Soroka, we trained 30 minutes in 3 sessions. And I'll go rather quickly because I am looking at the time. There is an issue with the Kinect camera with the accuracy of the presented elbow angle. We did some work following this experiment on improving the accuracy of the tracking, which I will not go into. I will just leave you, so we think that this lack of effect has to do with the issues with the Kinect representation of elbow angle accuracy. We did see, and this is the last slide that I will show today, we did see that patients that did not improve, and here on the right, patients that improved the TSRT more than the MDC, which is the minimal difference detectable change, meaning that there was a biological significant difference. Patients that did improve the MDC, we saw that they started out with slowing down their movement in the initial training session. And we think that this is an indication that they were actually concentrating in the screen. I mean, for the patients to improve in our trial, it's not about succeeding, it's actually about concentrating in the arm representation that they see in the screen in front of them. So we think that we do have a measure for patients that that were present in the virtual scene and patients that were not. And with that, I will thank you. I will thank the organizers for inviting me and thank my collaborators.
Okay, great. Thanks a lot, Sigal. I think we're kind of just about out of time anyway. I'm just going to take advantage. If you've lasted this long already, oops, that wasn't the, okay, yeah. If you've lasted this long already, then you probably really like motor control and motor learning. So if you haven't already received an announcement about this, there's a Zoom talk next week on reward-based improvements in motor control by Joe Galea. You can send me an email and I'll send you the link. I don't expect you to write down small details of here. And just on this note, to thank all the speakers. It was a great session, and thank again to the organisers for putting together a conference, and hopefully we'll get to see all of you in person in Accra next year. Thank you.
Thank you, Jason.
Thank you. Bye-bye.
Bye, thank you.
Thank you, Jason.
Bye.
Bye.
Thank you.
Thank you.
You're all welcome to the Gather platform, to the coffee zone, and then to the poster session. Bye-bye.
Bye-bye. Thanks.
Bye.